Okay, so here we're going to come in and kind of move on to part two of muscles here for us. One of the places that we're going to go here, and really what we're going to focus on in this part of the video, is how does contraction actually happen, right? So we've learned all of the parts from before, right? And the sarcomeres and the thin and the thick and the actin and the myosin and the troponin and the tropomyosin and blah, 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 blah. How does all this work? What we're going to do today then is actually figure out what is physiologically happening to make a muscle contract. And actually you might learn something surprising about what a contraction actually is. So from that end, we're going to begin with what we're going to call excitation, contraction, coupling. Now I'm going to link this video. I'm not going to show it to you for copyright reasons, but I'm going to link this video. I'm going to put this video up in your sapling so you can go back and look at it. It's just going to be a visual representation of the things that we're going to be talking about. So the first step in muscle contraction then is going to be this excitation, contraction, coupling part, and we'll talk about what that means in a second. And then we're going to follow it up with what we're going to call the cross bridge cycle. Okay. So excitation, contraction, coupling. What this means, and I realize that you can't read all of these little things on your computer screen or your phone or whatever, so we'll walk our way through it. Basically, what this means is we are coupling, right? We are joining the excitation part. So this is what happens when you get a EPSP, right? We're going to link that to the actual contraction part. We're going to take the neurological signal, right, the neurotransmitter, and we're going to convert that into mechanical energy somehow. This is what this particular little part of the phase is. So here's what's going to happen. We're going to make it very, very simple. Okay. Here we have our axon terminal, right, our little neuron. We have an action potential traveling down the axon, coming to the end and releasing neurotransmitter, just like before, right? And here we have a synapse, just like before, right? Only now the postsynaptic cell, the one on the other side, right, is a muscle fiber. It's not a neuron, okay? And so the neurotransmitter, in this case acetylcholine, right, crosses the synaptic cleft and binds to receptors on the muscle fiber. From there, we're going to generate an action potential, right? We're going to have electrical energy traveling in this direction and this direction away from the axon terminal. Okay, so right away, one important thing we've learned, right? For a muscle fiber to contract, it has to have a axon associated with it. Every single muscle fiber in every muscle, skeletal muscle in your body has a neuron that connects to it. They all have to be individually wired up. Okay, so there's a lot of wiring that goes on, which helps explain a little bit about why we have all of that connective tissue and all of that very highly organized bundling that we talked about in the first video, right? Because we have to have a place for all of these wires to go. So this action potential then travels away from the synapse here until it reaches a T tubule. Now you'll remember our T tubule is the middle part of a triad, right? And on either side, we have a sarcoplasmic reticulum that is storing calcium. So at rest, right, we've got a situation where we're actively pumping calcium into the SR, so we're requiring ATP to do this, right? And once the calcium's inside here, we're actually storing it right next to a little voltage-gated channel with a protein called calsequestrin. Cal, right, for calcium, and then sequestrum, there we go, calsequestrin, because we're sequestering or we're gathering and holding calcium right here. Now this action potential travels down the T tubule and hits these voltage-gated channels, and at this point, we actually open the voltage-gated channel, and all of the calcium we've spent all this time so carefully storing up It's released out into the cell. What does it do there? Well, once it's out, it's going to bind to one of our two accessory proteins on our thin filament. In this case, it's going to bind to troponin, right? And what happens here is similar to what happens in a lot of cases. When we bind to a ligand, our protein changes shape. And when our protein changes shape, it moves not only itself, but also it moves the other protein it's stuck to, which is tropomyosin. And when it does so, we do something very special. So at rest, tropomyosin is sitting on top of the active sites on the actin chain. And why that's important is that when the active sites are blocked, our myosin heads cannot make contact, right? Active site, right, is a place on a protein where we bind to a ligand. So if the active sites are blocked, our myosin heads can't bind to us, we can't form cross bridges. Right? And so the act of coupling contraction here with the excitation response is allowing us to form those cross bridges, because those cross bridges are going to be what actually causes our sarcomere to shorten. Okay. Now, if we want to go back to rest, what do we have to do? All we have to do here is we have to pull the calcium back into the sarcoplasmic reticulum, because once it's in here, we can't form cross bridges, we can't contract. So to rest, okay, we require ATP. We don't require ATP to contract, we require ATP actually to rest. Now, as far as the cross bridge cycle itself goes, I'm not going to get into all of the mechanics here, but again, this is going to be a situation where we require ATP not to contract, but to relax, right? And the reason for that is if we start over, let me pull my laser pointer back up, start over here at rest, we see the myosin head pulled back away from our thin filament. Now here, the troponin and tropomyosin have been removed, so we can't see them, okay, but they would still be here. And our tropomyosin would be covering these active sites here, right? But in rest position, we've got our myosin head, what's called cocked, it's in the cocked position, by binding to an ATP. And when we bind to the ATP, we actually split it off into ADP and a phosphate group. But nonetheless, we're still held in this resting position, okay? We release calcium, we move tropomyosin out of the way, and now the bonding force here, the attractant force here, causes the myosin head to swing down and actually form a cross bridge. Okay, we still haven't contracted, we still haven't done anything yet, but we've started the process. The actual contraction, the actual force that pulls the sarcomeres closer together, shorter than some anyway, is caused by the release of this phosphate. This phosphate is energy, right? We release it, and it causes the myosin head to pull, right? To literally reach out, grab, and pull the thin filament, and in doing so, it pulls the Z disc closer to that M line, does it on both sides, and so we shorten our sarcomere up, right? When we want to go back to rest, we have to take our ATP, we have to stick it back onto our myosin head and pull it back away and hold it away, okay, in order for this to happen. Now, like I said, I'm going to post videos here of both the cross bridge cycle and excitation contraction coupling so you can see them and watch them at your leisure. Those will go up on Canvas here shortly after this video goes live. Now, one thing that's important to remember is that just because we have a contraction, right, which is just the action of forming cross bridges and applying tension, applying force, it doesn't mean the muscle actually shortens, right? In fact, we can have situations where the muscle doesn't shorten, or we can have situations where the muscle actually lengthens. We're still providing force, we're still forming cross bridges, but we're still lengthening nonetheless. And so those kinds of contractions are called isometric isotonic and concentric and eccentric. Okay, so if the muscle does not change length, we form cross bridges, we're providing tension, right, which again is just the force of a muscle contraction, but we don't actually shorten, right? So if I used to use the example of going out in the parking lot and trying to pick up Dr. Diaz's truck, right? I guess I could still do that, even though his truck is at his house right now and I'm not in the parking lot. But strong as I am, I can't pick up the truck. And so I would provide all of my force. I would be straining as hard as I can, but my strength is not enough to move the load, right? If I can't move the load, the thing that I'm trying to move, my muscles can't shorten. And so I have an isometric contraction when the load exceeds the force. If the force and the load are kind of out of balance in different ways, we're going to get what's called a an isotonic contraction, okay, where the muscle does change length. Now, it doesn't have to get shorter, it can get longer. Both of those are isotonic. All that has to happen is the muscle changes length, okay? And you can think of these as being two parts of a weightlifting exercise, right? So if you've got your dumbbell in your hand, right, and you're doing curls, right? The first part of the exercise, where you bring your hand up closer to your shoulder, where you flex your elbow, right? This is the concentric part of that, right? The force that you're applying is greater than the load, and so the load moves and the muscle shortens, okay? But you don't just let the muscle relax. You don't just drop that weight. That's a really good way to hurt yourself, right? So you slowly let the weight back down, okay? You're still applying force, right? Still generating tension, but now the load is greater than the tension, okay? In a different way, right? Gravity and the weight is pulling the thing down, and you're resisting it, but not sufficient to hold it back up the whole way, right? So your muscle is lengthening even as you're resisting it. And this is again, an isotonic contraction, but this is an eccentric contraction. ECC versus CONC, right? So here, we've got kind of an example here of both an isometric and an isotonic contraction, here based around something we're going to call a twitch. Now, a twitch is what happens when a muscle fiber gets one action potential, right? This is not realistic. This is something you would see in a lab only, right? But this is going to be our base unit for tension, for muscle activity, for all of those sorts of things, right? In both cases, right, we're going to go through the same tension relationship, right, which is what we see up here. Okay, and what that is going to be is that there's going to be three basic phases through this contraction. So we're going to start with the stimulus itself. We're going to start with the initial action potential reaching the muscle. And it's going to take a little while for anything to happen, right? We're going to be in what's called the latent period. And the reason we have a latent period is because all of this has to happen before we actually start generating this, right? So there's a little period of time, not very long, but a short little period of time between the actual stimulus and the generation of force, the formation of cross bridges, all of that stuff. So the first part where we're actually generating force is called the contraction phase. Okay, force is getting bigger and bigger and bigger and bigger and bigger, and eventually it's going to peak out. And then we're going to enter the relaxation phase, where we slowly kind of turn things down, right? We go back to zero tension, zero force. And this relaxation phase is typically, although not always, is typically significantly longer than the contraction phase. And that actually becomes fairly important for us when we want to start having different levels of tension, stronger or weaker contractions. We're going to use that relaxation phase as a way to make that happen for us. Okay, so a little bit more about a twitch. Now we're going to use some slides here from my A&P2 course, just because I think they do a better job than the book of explaining it to you. So we're going to walk through this here instead. What we just saw on the last slide is called a myogram, right? Just this time versus tension graph, where we see how tension develops in the muscle and muscle fiber through that single individual action potential, right? And again, this notion here, this latent period, period of contraction, period of relaxation, different events are happening within here. And I'll leave you to read this at your leisure. I want to just kind of keep moving forward here. But again, the key important part here is that the muscle typically contracts. This initial contraction period is much faster than the period of relaxation, right? And this is really a more typical human muscle kind of myogram, right? Where we have a relatively long period of relaxation and a relatively short period of contraction. Now, different muscles will operate different ways, right? We can talk about fast and slow twitch muscles, right? Something you might have heard before, and we'll talk about kind of a little bit what that's doing in the third and final video. But really, ultimately, what fast and slow twitch means is basically just if we look at a myogram, how quickly do these curves occur, right? How quickly are each of these phases accomplished? So something like one of your eye muscles, right, has a really, really fast twitch, right? They all share the same latent period. All of that chemistry has to take place the same rate, more or less, right? But our difference here is going to be in the period of contraction and the period of relaxation. So something really, really fast twitch, like an eye muscle, our entire contraction is over really fast, right? Even before we've completed the contraction phase of a relatively fast muscle like gastrocnemius. Okay, now gastrocnemius is a big muscle. It's responsible for jumping and running and high intensity activities. Okay, but it's not the only muscle on the back of your calf. We also have a series of other muscles, things like soleus, that are not responsible for jumping and running and stuff like that. They're responsible for the maintenance of posture. And so they're going to be what we call a slow twitch muscle, where the period of contraction is very drawn out and the period of relaxation is very drawn out. It's not going to respond very, very quickly, although honestly, 80 milliseconds is pretty fast, but not going to be working at that same level of intensity as maybe a fast twitch muscle is going to do. So again, kind of keep this in the back of your head as we move forward into the next video for how the chemistry of that is actually going to work. Now, we're going to use all of this bit of information here in order to make a contraction of a single muscle stronger or weaker, right? And what we're going to refer to that as is a graded muscle response. Right, so just like we had a graded potential that was like a dimmer switch, a graded muscle response is the same kind of idea. You can have a strong contraction or a weak contraction depending on what the needs are, right? You wouldn't use the same amount of force to hold a kitten as you would to hold a brick, right? I certainly hope you would, right? And so the way that we're going to make this happen is through two basic ideas, right? The first one we're going to talk about here in the video, and move on to the second one, the next one, is we're going to change the frequency of stimulation. We're going to send, rather than one action potential, right, we're going to send several down the way, okay? The second one we'll cover in the next video here is we're actually going to change the strength of the stimulation. Well, like I said, we'll worry about that a little bit later on. Here, change in stimulus frequency, we're going to rely on the fact that we have this long relaxation period, okay? And the reason we're going to worry about that is that the force, right, the tension that's generated by a single stimulus doesn't necessarily have to start from zero, right? When we're totally relaxed and we have a stimulus, our force starts at zero and we add this much tension. Okay, but what if we didn't start at zero? What if we started somewhere here or there, right? And we added this same amount to it, right? Well, we'd have a stronger contraction, wouldn't we? And so that is actually what we're going to do. We're going to contract, and then in this period of relaxation, when we're losing tension but we're not at zero, we're going to have another twitch. We're going to have another stimulus, and we're going to add this amount of force to whatever force is left in the muscle. This is called wave summation, right? Sometimes we call it temporal summation because it has to do with the time, okay? But the idea here, and I'm just going to show you the picture, the idea here is that we have a twitch, right? And we add this much tension, and then we start to relax. But before we get to zero, we have another one. We add this plus this, and we get this. And we do it again, and we do it again, right? And eventually we get to a point where we generate an awful lot more force than we could with just one twitch. Now in this particular case, we've got periods of relaxation built in, right? So our myogram sort of looks toothy, right? This is called unfused tetanus. So tetanus, tetany just refers to a contraction. The reason it's unfused is that these curves are not complete, or not the same, right? You can distinguish one curve from the next, right? Now, if instead of having little breaks between our stimuli, we have them all packed very closely together, we get this, where you can't tell where one contraction starts, where one twitch starts and the next one ends, right? This is fused tetany or fused tetanus. And the problem with fused tetany or fused tetanus is that without these little periods of relaxation, we end up with muscle fatigue, right? And fatigue here in this particular context doesn't mean, oh, I'm tired, right? It means we physically cannot contract, right? So a muscle under fused tetany, right, will work until muscle failure. And we end up with zero tension at the end of it. So while we contract very quickly, we reach maximal tension very, very quickly, we end up working ourselves not to death, because obviously we can come back from this, but kind of the same idea. Okay, so we'll stop there. On the next video, the last video here, we'll talk a little bit about some of the chemistry behind why certain muscles are different twitches, fast or slow, and we'll look at some examples from the animal kingdom also about what's some cool things that happen with muscles there.